The thing is, we have to know about the vessels that do all of this. So first, we get a renal artery that comes into the kidney. That branches into segmental arteries. Then, segmental arteries go in between two pyramids. It's common to refer to the pyramids as lobes. So what term would mean between lobes? There's a name for the vessels here. Its name meaning between lobes.
Is it inter something?
Mm-hmm. Interlobar. The thing is, these little circular structures here and here are they're called renal corpuscles, but little lobes are called lobules. So here it's a vessel between lobules. So where it was interlobar before, it's now what? Interlobular. So interlobar means between lobes. Interlobular means between lobules or lobules. So the way that this pathway goes is renal artery branches into segmental arteries. Segmental arteries pass in between pyramids, becoming interlobar. Then, keep in mind that the outer or superficial region of a pyramid is curved in an arc shape. For that reason, when you get to the end of an interlobar, what would be the name of the red vessel curving here? Named after the arc pattern that it's following, they're called arcuates. So, when you get to this curving region of a pyramid, the vessel running along that curve is known as an arcuate artery. And realize that we see interlobar becoming arcuate here. That's exactly what you're seeing here. Interlobar becoming arcuate. So here we're just more zoomed in. Then, the arcuates start to branch out into what vessels again? Interlobular. Now, there's another name for these. These are also called cortical radiate arteries. Why does that name make sense? Because you are radiating out into the cortex. Well, this is where those afferents and efferents come in. If we glance over here specifically, let's say we trace blood up this interlobar, along this arcuate, then up one of these interlobular. In order to get into this corpuscle, there's got to be a vessel that goes in there and a vessel that comes out of there. What's interesting, though, is inside that corpuscle is a capillary bed, which is what we're zooming in on here. What do we call this capillary bed specifically? It has a name. It starts with a G. Glomerulus.
It's like glomer with something in it.
Ulus, yeah. Glomerulus. So, what's weird about the glomerular capillary bed, or glomerulus, is that it's supplied by an artery and it's drained by an artery.
Which is usually drained by a vein.
Exactly. So it's a little unique. You've got an arteriole coming in and an arteriole coming out. So if it's the same vessel type, how do we distinguish? Afferent arteriole, efferent arteriole. Exactly. So, sure enough, blood comes into the afferent arteriole, passes through the glomerulus. That's where it leaks the fluid. The fluid can then go through the nephron to do reabsorption, and then the blood leaves. Well, what was plasma then a second ago in this glomerulus becomes filtrate the second it leaks out and starts to go through the actual folded tube. The thing is, the pieces of the nephron then have names. We just called this part the glomerulus. The capsule that surrounds the glomerulus gets the corresponding name, glomerular capsule. But it also has a person's name that it can be used. Not glomerular capsule, but... Just learned it last night. Bowman's capsule, B. Okay, so glomerular capsule or Bowman's capsule. Well, then we know that eventually that fluid, now called filtrate, is going to go flowing through all of this. and work its way eventually toward this collecting duct. And that's where these vessels are going to reabsorb the valuable nutrients. So the first piece of the nephron is a highly folded tube, and it's at the beginning of the nephron. So when you're at the beginning of something, would you describe that region as proximal or distal?
Proximal.
Proximal. That makes this region then the proximal convoluted tubule. When that filtrate goes flowing through that PCT, as it's often abbreviated, it then will dip into the medulla and come back up in a structure called the loop of Henle. The thing is, the loop of Henle has a descending limb and an ascending limb.
Mm-hmm.
If we're tracing the filtrate going this way, what's this part of it?
Descending.
Descending, then it goes through the loop and comes up the
Ascending.
ascending limb. Then the ascending limb comes past the corpuscle again before folding again into the what? It's highly folded and it's now further away in the nephron. Distal convoluted tubule, or DCT. That eventually flows into our collecting duct. And here's the thing. Even though they're not showing it exactly correctly, these blood vessels here wrap all around these PCT and DCT.
Taking out the good.
Taking out the good stuff, exactly. This blood vessel here surrounds the loop of Henle, also taking out the good stuff, so to speak. That way, by the time you get to the collecting duct, does it contain nutrients and waste anymore?
No.
No,
waste.
just waste. The term for this capillary bed is the peritubular capillaries, because it's around the tubes. And this one is called the vasa recta. What does recta mean?
End? Bottom?
Straight.
Straight, right? Rect.
Oh, rect.
So this is given that name because it's a pretty elongated capillary bed with a lot of straight vessels. So vasa recta literally means straight vessels. Those are the ones doing that reabsorption, so that by the time you get to here, it's now just waste.
Now, is there any purpose for this dipping down?
Yes, but that's really more of a lecture topic that we'll eventually get to. And there, it's all about the fact that the deeper you get into the kidney, the more concentrated the osmotic pressures are. And that whole dipping down then into here actually means you reabsorb a lot of salt and a lot of water very efficiently. We'll cover that in lecture. We don't really do that in lab, so we'll get there in class eventually. All right, well, realize that we named mostly the arteries. Of course, the veins correspond. Where there was an arcuate artery, there's an arcuate vein. Interlobular artery, interlobular vein. Interlobar artery, interlobar vein. So the veins mostly correspond name-wise. So we've kind of looked at most of the things here. The last thing we've got to then zoom in on is this area over here and realize this chunk here is like zooming in on this chunk here. You're zooming in on a single corpuscle, where the word corpuscle is the term that means the glomerulus plus its capsule. So the whole thing is the corpuscle. Well, if you zoom in on one of those corpuscles, notice how the nephron, when it came back up, a piece of that nephron comes right up against the corpuscle. Well, that's this. That means that this piece here is not at the beginning of the nephron, it's toward the end of the nephron. That'll be important because we'll see that there's specialized cells in here, and it's important to keep in mind that that's toward the end of the nephron. So these two groups of specialized cells are together known as the juxtaglomerular apparatus. One clump of cells is found here in the afferent arteriole, and one clump of cells is found in that piece of nephron. These are known as the granular cells, or juxtaglomerular cells, and these are stretch receptors. If you have a stretch receptor in a vessel, what is it really measuring?
Pressure?
Blood pressure. Where does it make sense to measure blood pressure? In the vessel supplying the capillary bed or the vessel draining the capillary bed? Supplying. That's how you can tell the difference between an afferent and efferent arteriole, because only one of them is going to have a stretch receptor, and that's the afferent one. All right, well, why would it make sense to measure pressure in the afferent arteriole?
You don't want heavy flow going inside. You want to regulate what
So that
goes in.
is going to be feeling.
These series of cells here measure the stretch, which basically measures the pressure here, And the last thing you want is an overabundance of pressure in some of these vessels.
So is it interacting